Hello, everyone. This is Yamina from Dr. GPCR. I'm very excited to have back on for the second slash third time, depending on how you look at it, Dr. Arthur Christopoulos. Arthur, welcome back.
Thank you. Pleasure to be back. Yeah, I'm just waking up still, but it's great. Just tell everyone the truth. We just spent the last half hour talking about stuff before you press record. So we probably
Yeah,
should get we
yeah,
should
yeah. And I think we could have continued talking more, but it's too early for you and it's getting late for me, so we had to
yeah,
hit record.
that's right.
Thank you for coming back.
No
Today
worries.
we wanted to talk about your trajectory in the context of allostery. And there's a couple
Hmm.
of topics we left out, a couple of stories we left out last time. So you were talking about a Nature Drug Discovery review. How about we
Oh,
start there?
there. Oh, okay. That's in the middle
Or
of what I
we can we
yeah.
can start we can start from the beginning.
Yeah, let's start. Yeah, so I guess the key point I wanted to make, and I think I may have mentioned it last time when we were talking about just career and such in general, is to read outside your field and know your history. And of course, I'm an accidental allosteric person in the sense that I got tricked into allostery, and then when I did my postdoc, came back, started my lab. I was looking, but I was sort of kept it going on the side, and sort of ended up going full circle and becoming known for what I started doing. But as a grad student reading what I read, it was obvious that the history of allostery goes way back. Okay, there's first with enzymes and so on and so forth, and then ion channels and such. But even for GPCRs, it really started far sooner than people thought it did. If most people, sorry, look at the history. So if I just give you a potted view of what I recall when I was, doing my PhD, and I'm going to flag in advance that this is going to be rather muscarinic centric at the beginning, because actually a lot of the early work was on muscarinic receptors, mechanistically as well as much as anything else. But if you think about, okay, so the term itself, everyone know this, like 1961, I think there was a Cold Spring Harbor meeting where Monod and Jacob proposed the word allosteric for the first time. And at the time, Jean-Pierre Changeux was their PhD student and was doing the work. And so that's where the word first appeared, to my knowledge. And then in 1963, Jean-Pierre's first classic paper came out where they gave examples of allosteric enzymes. And 65 was the classic paper, the Monod-Weyman-Changeux model, where they first tried to formalize an allosteric mechanism on proteins. And that had fairly strict criteria. It had to be oligomers. They had to go between states. But that was indeed a two-state model. Funnily enough, I'm about to go off on a tangent. Del Castillo and Katz, who were studying the nicotinic receptor in the 1950s, they actually also proposed a two-state model for that receptor. It was one of the first two-state models proposed. And that actually is essentially the kind of two-state model that we look at these days, an active and an inactive state. That was on the nicotinic field. So they actually had a molecular mechanism of efficacy before we even coined the term efficacy in the GPCR field from, say, Stevenson and that. So back to where I was. All right. So an important year was, well, actually, in the mid 60s, you got guys like Arians, the Van Rossum and then later on Simonis and Van Rossum. They were theoretically, a lot of people know about Arians and his work on intrinsic activity and such, but as they were developing theories, and they were talking about the potential for what they then call the side receptor. And they also talked about even then interactions with side receptors. I didn't quite know what that meant, but they were theoretically talking about it. And so I think the Arians did suggest the term allosteric interaction around 66 for a side receptor, in principle, saying based on work that's already happening in enzymes. The same group later on coined the terms, I think, metaphonoid antagonism and metactoid antagonism, believe it or not, enough, because I remember Fred uses it in one of his early papers. And essentially what they were is what we would call today alpha, modulators and beta
Beta
modulators.
modulators.
Yeah, but that's actually, if you go back and look, that's what they were describing, even though they hadn't seen it. The first experimental example that I can recall, and was in 1969, and it was from Heinz Luhrmann's lab in Kiel, in Germany. And that's got an important connection to me and to my mentor, Fred, and you'll see why in a minute. They were basically looking at antidotes against organophosphate toxicity. right? So you got too much acetylcholine in the system. And they took nicotinic receptor antagonists, these alkane bisammonium compounds, and they started playing with the sort of symmetrical, or playing with, the appendages at either end of them. And they came up with a series that ended up inhibiting the effect of carbachol on atria in the heart, right? But once they plotted they did shield analysis on these things, they started seeing curvilinear shield plots, which was a dead giveaway of potential alpha type allosteric interaction. I think, and so in their discussion, they said, this could be something that Aryans called, allosteric. So that was so it was 1969, 68, 69. And the compound that they used, one of them was called heptane 1,7-bisdimethyl 3'-thalamutyl propylamodium bromide, or C7. That's the drug I did my PhD on, it turns out. There was a C6 derivative in the same series. That they ended up calling it later W84, W standing for Wasserman, that's the chemist that made it. And Klaus Moore and his group, and Evie Costini's trained with Klaus Moore, later on, they did a lot of work with that compound. So when I was doing my PhD working on C7, they were working on C6 essentially, but they were calling it W84. If you go back in the literature, they're all there. So take a breath. So 69, that was a seminal study. 68, 69, they did that work. What happened, and this is a history that people don't know, is in the early 70s, my mentor, Fred Mitchelson, who, as I said, really, really deserves a lot of credit, he did a sabbatical in Kiel. That's where he met Ullman and the team. And he actually brought some of these compounds back, the C7 one in particular, he brought it back to Australia, right? And Fred was at the time very interested in muscarinic subtype selectivity, because not much was known about how many subtypes there were and such. And he was looking at things like chemicalinium-3, which is an uptake inhibitor, and things like that to try and he was seeing some weird mechanisms. But in 76, as I said, this was the big book, the other big paper. I mentioned it in our other podcast. In 1976, he published the galamine, the neuromuscular blocker galamine, was allosteric at the antagonist at the M2 receptor, and he referenced the work that Ullman and such did, and he used similar analysis, and he was seeing curvilinear shield plots, the whole thing that now we would take for granted. So Fred did that in 76, so that was another seminal year, and that's why I mentioned it. And then I think, as I also mentioned in the other podcast, then things took off in the 80s, but particularly with the muscarinics. It wasn't long after that, so about 82, I think. Nigel Birdsall and Ed Hume and Simon Berg, they published their paper. It was Stockton, was I think the first author, would have been 82 in Mulfarm. So they did the radioligand binding assays, and really, if anyone does this stuff good, it's Nigel Birdsall and Ed Hume. I mean, they were the best, right? They validated, mechanistically and mathematically that galamine is allosteric at the M2 receptor. So we had the organ bath, the work of Fred. Then we had the work of Nigel. And then there's a lot of Freds in these. I was
gonna mention,
because we got Fred Aylett about to come up, and then later you got Fred Bruns. I mean, it's really interesting. So, and then of course, so all this work is going on. I hadn't even started doing research. I was just starting high school in the 80s. So it was the early 90s that I started doing my graduate work. But I think other important work in the 1980s were work that Fred Aylett was doing. Okay, so Fred's background, if I recall, was with Hank Yamamura and originally looking at GABA receptors. So Fred had already studied GABA A receptors and had formulated ternary complex models to describe allosterism and cooperativity at the GABA receptor. And then when he moved on to start his work looking at muscarinic receptors, he did a lot of really nice, there were a couple of seminal papers in the 80s that Fred published. I think one that really struck me was 185. There was one where he applied, it was more the ternary complex model as applied to G proteins. So don't forget, the endogenous, we didn't even mention that, that they're all allosteric because the endogenous allosteric modulator is the G protein, amongst other things. 88 was a classic paper from Fred. That was a theoretical paper where he really laid down a lot of the early, formalized a lot of the mathematical work around the ternary complex model, particularly with respect to cooperativity, alpha, affinity modulation, essentially. We had not gotten onto efficacy modulation yet. And then I would say 1990 was a big year for two reasons, and I think, yes. So, and it was because in the same year, there were two papers that were published in Mulfarm just a few months apart. Now, Fred Bruns would have mentioned his work at the A1. So that was the first, to my knowledge, synthetic small molecule, PAM, of agonist. Let me be clear, of an agonist. It was adenosine. And that was really important. I was fortunate to meet Fred many years later when they'd been taken over by Eli Lilly, and I visited Lilly in the 2000s, and I met him, and I could remember giving a talk. Nigel Birdsall and I were there, and I pointed to Fred. I said, "You guys don't understand who you've got over there, you know." This guy.
Yeah.
It was really important. So
I've had him on, Fred,
yeah,
and we talked about the bookends, which is really phenomenal.
Fred. So Fred Bruns, I mean, Fred Aylett, I've mentioned before, and he did some incredible. You should read some of his GABA papers as well as his muscarinic papers, and he even spoke talking about inverse agonism and stuff like that. And I think a lot of Fred Aylett's work was influenced, and a lot of other people's work, by people. A guy like someone like, I remember, Gregorio Weber, and also Mono himself. They did a lot of work about protein equilibrium, circle wheels within wheels, and that. Anyway, 1990. So that's a seminal paper, the Bruns and Fergus paper, that wherever they discovered the first synthetic. And I use the word synthetic on purpose because a few people have called me out on this before. There were hints in the 80s of other endogenous allosteric modulators. There's a tripeptide that some people have done work on. There was a couple of others, but they hadn't been mechanistically validated enough. But there was evidence that there may have been. But I put them in the endogenous category. And actually, we wrote a review in 2015. You can find it all about endogenous allosteric modulators and GPCRs if people are interested in looking on that aspect of it. But in terms of synthetic drugs, it's not there. So that was the Bruns and Fergus paper was key. But actually, a few months before that, there was another paper published by Stanislav Tucek, the Czech Republic. Now, he is another important figure. So Jan Jakubik was probably the best known person from his group that trained with Stan. But there's another guy called Jan Proška. So they published, actually a few months before Fred did, the first PAM of an antagonist. And that was alkyronium at the M2 muscarinic receptor, was an antagonist, a PAM of the antagonist tritiated NMS. That came out before the Bruns paper came out, but they were just a few issues apart. That was the first time I saw a PAM. And again, it was the M2 muscarinic receptor. But if you go back and look at the two papers, two interesting observations you'll see in both is the curve goes up. It's like a bell-shaped curve. The PAM effect goes up, and then at high concentrations, it comes down. Now, in both papers at the time, it was interpreted that the down phase is due to a second interaction at the orthosteric site, right? High concentration. So they said the PAM at lower concentrations it's a PAM, and then at high concentration it hits the second site. That's not necessarily the case. Certainly with alkyronium, it was shown it was a kinetic artifact. And we're going to come to that in a minute, because that we've done some work on that, but it was a kinetic artifact. And the bottom line is, in fact, I'll tell it now because it's the next really important paper, 1994. It was again from Stan Tucek, right? And this time with Jan Proška, and they looked at the mechanism of alkyronium, and they showed that the down phase was actually a kinetic effect. In other words, the system had not approached equilibrium. Because if you think about it, if affinity modulators will affect the dissociation rate and or the association rate, or the slow them or speed them up, right? And if you slow them, the ratio of how much you slow or speed up, right, is going to determine what the affinity effect of the PAM is going to be at equilibrium. So when you talk about a PAM or a NAM or whatever, you were talking about its effect at equilibrium, not anything else. And it's also contextual in the orthosteric ligand, because that's probe dependence, right? When we talk about. So it turns out that obviously if you slow the kinetics enough of the radioligand, right, you're not going to be at equilibrium when you stop the assay. That's why it drops. And if you let it go for longer, and in fact, they did that. And in 2004, we actually published a paper in JPET where we looked at that in more detail. You look for PAMs and NAMs at the muscarinic receptor, and you can see bell-shaped curves and biphasic curves, right? Bell-shaped curves, all biphasic curve, competition curves, and all due to kinetic artifacts associated with modulators. And you can change that if you get the assay conditions right. So that's an example of where a more complex mechanism isn't needed. The simple affinity modulation can explain it, but you've got to think about kinetics. And a lot of people now, of course, are always thinking about kinetics. Yeah. So that was important. Those two papers were important in 1990. It was interesting. I saw similar effects. The 94 paper, the other thing about the 94 paper, and this is why I bring it up, from Proshka Tuchek, Stan Tuchek, to my knowledge, because I hinted at this in one of the other podcasts, right? That's the first time the word allosteric was ever used.
Interesting.
So go back and have a look at that. I can't find any earlier references to it, because even in the earlier papers, whether it were Nigel's group or even Stan Tuchek's themselves in the 1990 paper, they talked about everyone talked about the classic binding site, the primary binding site, analogous to the substrate binding site on an enzyme. Yeah, even in the Shangzhou paper from 63, they were talking about substrate binding sites and allosteric binding sites, but they proposed two models, and one of them looked like what now we would consider reciprocal interactions between two sites, but there was another one which was so indirect that didn't look like it was reciprocal. And later they used the word isosteric, and they were talking about the bonds that need to be formed within the primary binding site. So the terminology can get confusing, but allosteric was never questioned as in other site. And 1994, that's I'm telling you, if anyone can find an earlier instance, I'd be very interested. But that's, so we owe Stan Tuchek a lot
yeah.
And then basically through the 1995, Nigel and Sebastian Nazareno published a classic paper in Mollopharma. They just went to town. They went to town on all these mechanisms of allostery known to date and all the analyses and models and such. So there's a lot of modeling starting to happen around then. In the late 1990s, basically there was two things to note. One is the area of molecular biology had come in, and so people, again, in the muscarinic field, because they had some tools, were starting to do structure functional mutational analyses. And two people I need to particularly highlight there, John Ellis, and the other person was Issam El-Fakahani in Minnesota, who I ended up doing my postdoc with. So they were doing a lot of that work around then. And also around that time, Klaus Moore, who had trained with Lulman back in the day. So Klaus was doing a lot of work, started doing a lot of the work in the 90s onwards, I think 80s actually, and his lab. So Evie, as I said, is a contemporary of mine. So she trained with Klaus. I trained with Fred and Klaus knew each other. And I was fortunate because in my PhD thesis was examined by Stan Tuchek and Klaus Moore. Right, that's So Fred sent it off. That's how he works in Australia, and they came back with no corrections. So I was pretty thrilled. Yeah. That was really cool. So the other thing about the late 90s is, and this is where things really started to take off, a lot of work is then being done on the Class C GPCRs, particularly in pharma. So there were a couple of important papers that came out in the late 90s from Jean-Philippe Pan, but I've also some people at Roche in particular, in Basel, and when Jeff Conn was at Merck, Craig Lindsley was at Merck. So they were doing a lot of work on mGluAs and the other Class C. So a lot of people think that's kind of where it sort of started, because the industry, and then what happens around 2000, 1999, 2000, that's when everything flipped. Pharma went from biochemical radioligand binding of assays to cell-based functional assays. And everything started coming up. They had no idea what they were seeing. So that's a seminal period right there. So a lot of people would sort of think the late 90s, the 2000s, the farmers changing the way they screened. That's a lot of stuff. Because if you look 2000 onwards, it just goes out of control, right? But actually, if you look, it was always there. That's why I think you just have to know. Yeah. And in fact, I mentioned how I met Terry when I was a grad student. I mentioned that last time, and it was 96 when I met Terry, just before on my first trip overseas. He had dabbled a little bit in allostery in the late 80s, because I think, but I'm looking at muscarinics, I think it was looking at galamine actually, but I suspect because they had a respiratory program. But Terry's work really, I think, was when they started probably doing chemokine programs at GSK. I think that's where Terry really got into all the aspects of allostery and the craziness of it. And we can talk about how bias later. Yes. So and I think the 2000s is where it just took off. And for me, and now we'll come to that review that you're talking it's one of those weird instances where I did my postdoc, my PhD. I did my postdoc, and we had the discussion about, well, do you come back and do?
Yeah.
And I had enough people supporting me or encouraging me to stay with this receptor theory or pharmacology stuff that I liked. Terry being one, Paul Insel, a bunch of them. Rick Newbig, as I mentioned last time, I think. So when I came back and started my lab, I started my lab looking at cannabinoid CB1 receptors as potential pain targets, and we were looking at, and so I was diversifying the sort of the receptors I was looking at. I did a lot of work on 5-HT2C. We were the first lab actually to publish that showed the 2C coupling to MAP kinases and the effects of RNA editing on that, not on RNA editing itself. So on the side, I had this. So this is one of my aha moments. I know you talk about aha moments.
Yeah.
All right. So I didn't discover, allosteric or anything like that, right? The aha moment for me was the realization that this is on every GPCR. It has to be, right? It can't not be. And then the decision is, well, how the hell do you go around proving that? It's a lot, right?
Yeah.
And so I just made the decision that I'm just going to do it somehow. So while I was doing the main sort of work that I was studying at the time in the lab, and I brought back the cell lines with me from the States and everything, and I'd go in on weekends and just start doing more experiments on my own on muscarinic receptors initially. And then as I would be getting in more students and postdocs and such, I would basically say, okay, different receptor, all right, do it that way.
Yeah.
And that's how I launched careers. So I said, okay, where am I looking? And then I looked at the AA1 work. There was some because of Fred Bruns's work. There was adenosine modulators out there, the PD compounds and such. And at the same time, I met my second longest collaborator, who's Peter Scammels, who was the head of medicinal chemistry in our institute. Now he's still there. He's an associate dean research. And Peter had a chemistry background, adenosine chemistry background, because he had worked with a guy called Ray Olson, who in Florida, who had worked with Fred Bruns as well, I think, or they collaborated. So I said, oh, there's a chemist that can make these compounds. So I went from muscarinics, so I would keep them bubbling away. And then I went to then I started that's how I went on to the A1 receptor. So that's how, and I remember Jim Black saying, you got to have the chemistry. Because I was also, that's around the time I met a lot of people. And when I wrote to Jim, and Jim was at the beginning, early on, did not believe in allosteric modulators and exogenous or synthetic, because his view was the chemistry has to be there, right? If the body makes its own things, and that's agonists and antagonists, yes, but later on we had chat, and when I showed him that the chemistry was there, but that's why I learned, okay, I need chemistry, and then I switched and I need receptors, and then when Katie Leach joined my lab, for example, I know the timelines are jumping back and forth a bit.
Yeah, I can tell, but
this is all in the 2000s. This is really important. So, Katie came to my lab and as a postdoc, and she said, I want to work on a receptor that has clinical relevance. And I looked around, and there was only one GPCR that I could see. There was PAM on the market. It was discovered by accident. I know it's Sensipar. The drug is cinacalcet for the calcium sensing receptor. It's hyperparathyroidism, as a consequence of chronic kidney disease. It was discovered by serendipitously, right? By Ed Nemeth when he was an NPS, and Ed's got a funny story to tell about that. But I said, okay, I guess I'm getting into class C GPCRs now, because there's a PAM on the market, and it's a calcium sensing receptor, and no one has done the molecular pharmacology on it, but it's basically been looked at by endocrinologists and such. So back go, and then that's how Katie started working on class C GPCRs, and so it's boom, 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 boom. So that's how I ended up doing a lot of different receptors. And the flip side is I also collaborated a lot with other people working on these things, because I sort of became known as the go-to guy. And this is right. Sorry, the review. So 2002, you can almost draw a line in my career before 2002 and after 2002. And it's one of those rare instances where I got known more for writing 2 reviews in that year than I did for the primary research papers I did beforehand. Right? It just is what it is. They were being published in journals that not everyone would necessarily read. But I got asked by the Nature Reviews Drug Discovery. So the first year of Nature Reviews Drug Discovery, like the 3rd issue or something, I got asked to write a review on allosteric modulation for receptors, not just GPCRs. I don't know how they found me, but I was delighted. So I wrote, that's my first sole authored review on that. And then The same year, I wrote a big review with Terry in pharmacological reviews, right? And that started from in 2000, Terry had asked me to write a chapter for the current protocols in pharmacology, and I started doing that. And then after a while, it's got so big, I wrote back to Terry, I said, you know what? There's all really cool stuff in here. Why don't we combine this with some of the stuff that you're thinking about and put it together? And then there's a really big review. So 2002, I published 2 big reviews, right? The Nature reviews and the Pharmacal reviews. And after that, all of a sudden, people just sort of knew who I was, right? So encapsulating the work, but so and then everything just took off. So there was so as well as building up my own lab, there was a lot of work going on in terms of, I was being contacted by pharma. So I got to see how they're seeing it from the inside. And there was a lot of stuff that I learned a lot from there because they needed to learn a lot. I realized I had other colleagues collaborating with me. So that's how I got involved with the CB1 PAM stuff that was published in 2005. I mean, that's a funny story because I remember in 2004, Organon invited me to a meeting in Edinburgh. They were hosting, and I spoke on whatever I was doing at the time. And I remember seeing one of their posters. This is actually an important story too. I actually see one of their posters, and on it they had they were using luciferase assays then for cyclic AMP. And I looked at these, looking at these compounds, these CB1 compounds, and I was looking at their dose response curves, and I was going, this is impossible unless. So that basically they were collapsing the Emaxes.
Wow.
But if I looked at but I'm looking at them carefully, and I'd say the EC50s are going to the left, and I'm looking at this. I go, all right, you've either got error in your assay. If this is true, I said, these compounds must be increasing affinity while they're decreasing efficacy at the same time. Like, I go, so that's just from this little panel on a poster, right?
Yeah.
So I thought, so I didn't forget that. Then I went back to Australia, and then I got an email from Ruth Ross, who at the time was in Aberdeen. Now I think she's in Canada, and Roger Pert, they were collaborating. And Ruth emailed me saying, look, we've got this collaboration with Organon, and we've got these compounds, and she'd done binding assays. And she goes, we've discovered the first PAMs of the CB1 receptor. And I saw the curves, and they were going up, right? Yeah, they were going up. And I realized they're the same compounds, right? And I said to Ruth, what did they do in function? And then they looked at Vas deferens, and they looked at gamma rest, and they're like, what? It collapses the curves. I said, no, they're not. They're PAMs of affinity, but they're NAMs of efficacy. That was the first example I ever saw. And that is when I came up with my first operational model of allosterics, because I had to explain it. And I had, alpha and beta. Up until then, the models were all based on alpha.
Yeah.
So 2005, Price et al. Molecular pharmacology. That's why. So I only come up with models when I have to. The data force me to. I don't have time to. I used to when I was young, simulating whatever. But that's how I came up with it, because I had no choice. I had to try and explain it. Same year, Terry published essentially the same model in a review. And the same year, Fred Aylett published the same model in JPET. I don't know why, but in 2005, 3 papers came out with the operational model with an alpha and a beta, right? Yeah, and it wasn't until 2007. I know David Hall also published one in 2006 that had efficacy in it as well, I think. Yes, it was a book chapter. 2007 was where we were doing more work, and we were seeing not only alpha and beta effects, but we were now starting to see allosteric agonism too, right? So it changes your baseline going up. So I'm looking at that, oh no, we have to put efficacy allosteric agonism in as well, right? So that's when it came up with the operational model for allosteric modulation and agonism. So the tau beta effect for a modulator. That's why we're seeing it. And I think, again, 2008, I think Terry published the same model sort of thing. David Hall published. But again, mine was driven by data. We had data and I couldn't explain it, so I had to do it. So that's how we did that. And that was sort of my good way to sort of models, my approach to models. It's, they can be too complicated, and I want models that can be fitted. And since then, a lot of people have done far more elaborate work in, there's David Hall, Jesus Geraldo, Sam Hall does a lot of work with the kinetic aspect of things, right? But so my criterion has always been fitted to data. It's got to be fittable routinely to at least quality data, because you can make more complicated models and simulate and see how things could go. So as long as it can be, so I always try to find the trade-off between mechanism and empiricism, parsimony versus heurism. That's my approach. Unless the data call for it, I don't have time to do it. You know what I mean?
Yeah, but when you think about it, isn't it beautiful that in cells in nature we see these events and then you can fit a mathematical to put numbers to it? I mean, I think
Yes,
it's just beautiful.
I, 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 well, this is James Black was, he coined the term analytical pharmacology, and it was specifically to describe this approach, and that's hugely influenced me. And then so I essentially, because I was fortunate. I told you how earlier on I corresponded with Jim, and it was because I started getting involved with NCI-UFA and nomenclature. We can talk about that later. And then in 2007, I got to meet him, and by that stage, we had a lot of data, and he was fantastic. He picked me up at the train station. We went to Dulwich at his favorite pub, and we had a fantastic lunch and spoke about, and I did the whole fanboy thing, how much it meant to me and how much it's influenced me and how I'm using the operational thing, but I'm applying it to Alice theory and And it was great because that's fantastic. And I believe you because there's enough chemistry examples now in modulators, right? Sort of thing that he was, yep, it's there. Then he floored me with one question, and that's haunted me to this day because it's influencing what I do now. He goes, "That's all great." He goes, "By the way, tell me, how many clinicians are you working with?" And I just like my jaw just dropped because at the time the answer was zero. Right?
Yeah.
So I'd go from a receptor. I need receptors with examples. That's why, and that's how I jump. But then I need chemistry. Then I go, well, now I need animal models. And subsequently, post 2010 and such, you'll see. But then we got into structural biology,
Yeah.
computational biology, and that's how I started working with Brian Kabilk and Shoy Kep and Ron Draw, and now we do work with Yin Long Miao and Jens Karls. That's how I've met all of those guys like Roger Sinahara. But whatever technique was called to answer the question is how I approached it, right? And that's, I think, the way you should approach it. Don't be a technique person, be a question person. But when Jim Black asked me at that time, how many clinicians am I working with? The answer was none. And then now, of course, I am working with clinicians. Say, well, what do you see? And then I try to go backwards as well. But that was a really formative stage from between 2000 and 2010, both for me and for I think people that I trained. And I just collaborated with everyone because if I didn't have it, like the CB1 is a good example, right? I didn't care. I was like, I haven't seen anything like this before. Let's do it. There was another paper I recall on the prostaglandin D2 receptor. That was with Evie Costinus. And I think, yes, turns out that their compounds weren't allosteric. They were just slow binders. So we showed that it was kinetics, not whatever. So I'd collaborate with anyone because I didn't have the resources or all the stuff at the time. And that was really good because, you got this balance between leading and collaborating, and you need to do both. But also it felt really good that I stuck to my guns, all the early advice I was given. I didn't go down the traditional path of becoming a neuro guy or a cardiovascular guy or whatever. I just used allostery as a mechanism to train that become neuro or get cardio or whatever folk, and then of course, as the collaboration with Patrick progressed, we brought a lot of the techniques in-house, and we do a lot more chemistry and a lot more animal work ourselves, all the structural biology ourselves. And sorry, and no, it's that was me. And the other bit, the pharma connection was really important. I still got to see a lot of stuff in pharma, and now they're shutting down programs just when I was saying you're not doing it right, but now they're coming back, and we'll see what happens, because now we'll see how much the rational knowledge or the rational design of allosteric modulators, based on the sort of work that we did, because we helped inform structure activity approaches and such, and we'll just see how many of those come through now. You'd like to think more than before the serendipity. Up until then, it was all serendipitous.
That's a whole.
Yes, that's a lot. Yeah, and since then, you can see how just it's expanded, and lots of people are looking at it in so many different ways. The other thing I think I did that was important in that time frame, I got involved heavily with NCI-UFA, and the nomenclature work, and that was really cool and key too, because that introduced me to a lot of other people that I didn't know, and also I got to see a lot of high level stuff, other scientists, great scientists, from other fields, other receptors, and the dialogue there. Now Steve Alexander is the chair. I'm still deputy chair, but I've hardly been around since I've been sick. So, and there must and such. But that was those days with NCI-UFA and Michael Spedding was fantastic as leading it. That was really important. I met a lot of people, and it was around that time that I also, not long after, I met Jean-Pierre Changeur, and that was a real thrill for me to meet, Jean-Pierre. And I gave a talk at the Pasteur Institute, and in 2016 we wrote a review together in Cell. That was a highlight for me to write receptor. Allosteria is like a unifying mechanism across all receptor families, and that sort of started with the NCI work first, making the connections, seeing what the other receptor, the fields, nuclear hormone receptors, ligand voltage gated ion channels, and the bilcaterals and such, for instance, receptor tyrosine kinases, as well as GPCRs. And I'm sitting there going, all right, I can see similarities and differences, but mostly similarities, including the way we approach allostery and what we call it, and the way we approach what's now called biased agonism and what we call it. And that's what I thought, well, we really should do something about trying to put this together. So that's how I got to meet a lot of these people, and I'm so grateful that I did. But we also then were tasked. I don't know if you want to. Do you want to ask about?
I do want to stop you for a minute, because a couple. I didn't want to stop you because all of this is fascinating. One, number two, when I think about these concepts, when I was learning them, these concepts were known. So just to hear that these things were actually not known, but they had to be discovered, you needed the tools to actually measure this, and then you need to create a model that explains the event. Is just phenomenal. And then you mentioned that the Institut Pasteur and Jacques Monod and Changeux. I learned this during my undergrad. Just to think that I actually know someone who wrote a review with Changeux. It's such
how do you think it was for me? I mean, I was a student. I started reading this stuff, and also when I was working with Fred, and this is the problem I was trying to allude to earlier. At the time, a lot of people were thinking, it's rather esoteric in the sense that it's probably just your receptor or something like that. And that's why I said my aha moment, saying, no, I think it's everywhere, and we've got to prove it, and we just got to buckle down and just do it and take the risk. Because the other aha moment for me was around 2008, but the bitopic ligands. So that is a word I can say I did come up with, bitopic, right? They're basically hybrid molecules where you take an allosteric and an orthosteric pharmacophore and you link them. They're a bivalent ligand. So Klaus Moore and his group are doing a lot of work in that space too. And again, it's how things overlap. They were calling them allosteric, and we called them bitopic. And so, but the way I stumbled onto it, it shouldn't have worked. And it was when Celine Valente joined my lab. There was a muscarinic agonist, McNeil A343, which has been around since the 50s, right? Fred did a lot of work on that, and it was subtype selective in its agonism, partial. And when I looked at it, I go, well, there's methylammonium bit, which is essentially like acetylcholine, and this other bit. So I just had this weird idea, and it shouldn't have worked. We reverse engineered it. I basically said to Celine, because Celine had chemistry training, and I said, can you chop this molecule in half? And she said, why? I said, I remember my exact words. I said, just humor the old man. And we did. You can find it's Celine's 2008 JBC paper. And that's where I came up with the bitopic model, the mathematical model too, right? For it. And we used it to analyze that data, but also I collaborated with Klaus Moore on their data. So we reverse engineered it and found that one part of the molecule acted as a NAM of the other part of the molecule when we combined them. Now, you can talk to Brian Shortcut, he'll tell you that hardly ever works. Reverse engineering, let's just fly away, That was a fluke. That was a fluke, right? But that's how I stumbled onto the idea. And then I started looking at how many other extended molecules out there that have functional selectivity or whatever are because of this. And at the same time, Klaus, I was working with Klaus and collaborating with him in his lab, and Evie was part of the collaboration too. They were rationally designing these things. They were taking an orthosteric pharmacophore. And I mean, we did that subsequently with Peter Scammels, but they were doing it. And then again, I said, well, I did some modeling to say, you should get one of your PAMs of an antagonist or whatever and combine it with this. And that came out, I think, a year later, 2009. And that's what they call allosteric. But that's where the whole bitopic thing started. So you either rational design them or you reverse engineer existing things, and you might find that that's the thing. But more often than not, and we've got a story that will be coming out when we finish writing it this year, where some of our design, you can definitely, so in 2014, we actually designed bitopic ligands that became biased agonists of the A1 receptor. And we're looking at those for heart failure with Lauren May now driving that work. And we actually have the structure now. And it doesn't bind the way we thought it would bind, but it works the way we'd like it to work. So, if it gives you a different conformation and gives you bias, that's what we want at the end of the day. And that's one of the things, if you can make them drug-like enough, by topically, it can be really useful compounds and probably biased as much as anything else. So that was another aha moment. But yes, and then the Sean joke. All the rest is, I don't know. It's all a blur after that, because you get invited to meetings and you consult with drug companies. And again, I remember the earlier 2000s. The reviews had come out, and I'd be going to conferences and stuff. And still, the first ever GPCR Keystone meeting, I remember just presenting a poster on some of this stuff, whatever. But then after that. But I was going to the FASED meetings and all these things. And specifically, I would be targeting drug company posters because I wanted to see what was coming out and where there were the hints of this there. And the other thing I would often do is especially after the review, sometimes I'd cover my name when I was talking to people. So I didn't want to distract them. But if they saw the name and they go, oh, because I'd rather like a freer conversation sometimes, because it goes either way. You know what I mean? You know, people thinking up. They didn't know who I was, so it's better just to have a free conversation. Actually interested in your work.
Exactly. They think this guy is super interested, and I'm talking about my work, and we're having a fun conversation, and not necessarily knowing who you are.
But through that, that's how I got involved in a lot of pharma consulting, or just got some contracts, got a lot of collaborations, and seeing it from their perspective was really, really interesting, because you could see what they could do, but you could also see where some of the things that they weren't doing. And so I guess there are some funny stories from those days. But
it's an interesting point that you make, about seeing what pharma and biotech are doing and the way they're doing it. And I think unless you get the opportunity to see it, you don't know. You can hear people talking about it, and it's a fascinating world. And there is a huge difference between big pharma and biotech as well.
Yes. So. And a challenge at the time. I mean, well, we'll come back to the 2000 the NCI UFA review thing in a minute, because it came out in 2014, right? But we'll come to that in a minute. The interesting thing about the pharma stuff that I saw was, on the one hand, we would be talking about stuff and how we would analyze or do the experiments, and the easy thing to come back is, well, that's easy. We got to screen hundreds of thousands of compounds, and we got to do it in an X format and X whatever. So it's fine and dandy to have X dose response curves and then do whatever. So that part of the trick was just finding a middle ground where, how can you design, and that's why I prefer models that can be used to analyze titration, whatever, but knowing the limitations versus the advantages. But also things like, well, okay, if I was in there, if I had a 384 well plate in front of me, how would I redesign what's on that plate to be able to extract the information that I'd like, as
faster than having to run 1,000 compounds.
Even things like, that's an example saying, right, rather than running replicates, if you ran just singles, right, but they were dose response curves, and you globally fitted the data, right? Even the odd outlier, doesn't matter. You'd still be getting more out of it than if you had lots and lots of replicates and just to do one, whatever. Yeah. That's one example. Another one, is
that's assuming you have a robust enough assay.
Yeah, exactly. In fact, let's assume that's all in the case. It's all been done. It's just the same assay. It's just how you redo it. Because your point about having a robust assay is actually a very good one, because another really interesting example and story I had was from around 2004 or so. I was visiting one of the big ones, and at the time they were doing a lot of gamma S based assays, and the signal to noise in gamma S is not the best. So there they would use a lot of replicates, even for the East, and then to do any type of titration type experiment, which was fair enough. But I remember giving a talk when I showed, and this is a curve shifting to the left and curve shifting to the right, and I just pointed out EC20 and EC50 and EC80, right? And I go, so where's your window, right? If you're right, okay, where's your window? So when I pointed to I remember pointing to an EC80, and I said, okay, if you got a PAM, for instance, I go, where's it going to go, right? Because right? You hit the ceiling, right? So all you're going to be seeing are flat lines. I looked out in the audience and I saw a whole bunch of people slap their heads. It was like, and I'm, oh, this is how they're running their essays, and they're asking me why they can't find PAMs. For, you know what I mean?
Yes, because you can't run it with an EC80.
Yeah,
It's 20
yeah,
to
yeah,
40 max.
yeah, that is taken for granted, right? But even back then, it wasn't, because I remember speaking to some other people that are really, they're better than me, and but they were saying we're not finding allosteric compounds. And I go, well, how are you screening for them? And we're just adding them in the plates. I go, well, how do you screen for antagonists? Or we do titration experiments, we do an EC something. I go, well, you should be doing that. They weren't even doing that back in like 2002 or something like that. The big, a lot of the large farms, I don't want to paint everyone with the same brush, but it's, you take it for granted, but even, and it changes very quickly. I think biotech, and the smaller companies were moving quicker. But yeah, things like that. That's the sort of stuff I learned. So in terms of the review, back to the thing.
Yes, because let's just give a little bit of context here. So before we hit record, I asked where I actually came up with. I've been thinking about this for a long time. What would that look like if we brought together everyone in the field who has been there at the beginning, and to write a book about the history of GPCRs? And now, as we hit record, I was thinking about it, and I think it should be a conversation. So I need to think about who to invite, and we can gather these stories and use these stories and build them into a book. But I think the conversation would be more pleasant to watch and to participate in. But I'm putting a pin in that, and that's when we talked about writing.
And that's right, because that triggered my thoughts, because I asked you how would you do it, because here's a story that happened to me about, and it was my NCIUFAR days, and I mentioned earlier about the committee and the other, and I thought we need to try and write a nomenclature document, or we were tasked to write a nomenclature document on allosteric ligands, and that's virtually impossible, but you talk about herding cats. I had to try and get so many leaders from all the different fields, and we all then had to talk the same language and agree to different things, and then realize we can't cover it all. But that's how I got to meet Jean-Pierre Changeau and Bill Catterall. So anyway, we did it. It was published in 2014, but it started around 2007. It took me 7 years, basically, to finally get that one over the line. And it'll have its critics, but it's the best we could try to do to try and standardize things. But it was an interesting experience, and I met a lot of really great people and scientists. But that was 7 years to get that one. Then around the same time, what they were saying to us after it was done, actually, the idea was, okay, we got to do one for bias now. And at the time, I said to me and Rick Newbig to try and pull that one. And I was like, oh, geez. I said, I'm never doing this again. So, and again, I mentioned this to you before we started recording. So my proposal for that one, which we never did in the end, and later on, there's been a more recent one that Dave Gloriam did with IUFA that came out in the BJP. But that's a different format to what I had envisaged for the original one. My idea at the time was, we're never going to do this 7-year thing again, right? And it goes back to how you're going to do the book, right?
Exactly.
So I had suggested that we should do a 2 or 3 day symposium. It just, we couldn't find the money at the time, but I said, do something like that Cold Spring Harbor thing that they did back in the day with, Mono and such. Yeah. Get all the people, the leaders, get Michelle, get Bob, get Brian, get Rick Newby, get I'll be there, Terry, just Laura Bob, get all these people that were sort of really in the thick of bias at the time, and basically do the symposium and use that as the goal being, we're going to come to an agreement about what we're going to be, how we're going to be calling these things, and then we go off and do the review, whatever. That was the idea, but it never came about. But I think that's one way of doing it. The other way is what you just said now. The other way is conversations, but getting them and herding them and whatever.
I think from a convenience standpoint, a Zoom conversation is easier, but I like your idea of bringing everyone together you know, 2 to 3 days locking it and being able, it's as a starting point, bringing all of these people together for 2 or 3 days and having these conversations. One, it's a pleasant experience for everyone because everybody knows everybody at this point. But also it's a fun thing to do and it's a nice starting point. I think for my book idea, I would need an outline and then have defined chapters with defined topics and find people who I can interview and we can talk about those and then put all of this together.
Well, I think that the symposium idea has been attempted in the past. I remember it was 2005 at an ASPET meeting at Experimental Biology, and it was Richard Mailman at the time who wanted to convene a symposium around bias. Well, we're calling it functional selectivity, right? And such.
Yes.
And I was invited to speak on that at that. I couldn't make it in the end, but my talk was I was unwell, so Patrick presented my talk for me, I think. But then after Brian spoke about some stuff, there was a bunch of authors, including Michelle, there's a bunch of authors on it, Mark von Zastra. And then afterwards, we were all asked to write a review for JPET based on that symposium. And it's Urban et al., you can find it. It's a highly cited review. And that was on functional selectivity. So that was an example of a first attempt at doing something like this. But that whole, I mean, that's a segue into bias, which I'm not going to talk about the history of bias other than that's another example of where just things intertwined. Just very briefly, for example, there was a review that Brian Roth published in 1987, I think in Life Sciences, where they suggested antagonists could have receptor effector specific effects, I recall. So that was like a hint. I don't know if he was thinking about bias at the time, but I'm pretty sure now he'll say, I know Brian is, and it was, yeah. But again, Richard Mailman, I had mentioned in the early 90s, was calling it functional selectivity on the dopamine receptor, but I'm pretty sure that he was thinking about bias, what we call bias now. Terry, there are two reviews that Terry published in 1995 in Tips that people, if you haven't read them, you should read them. There was a part A and a part B, all right? And he was really early on bringing together the idea of the promiscuity of GPCRs and the pleiotropic coupling. And then the first review sort of focused on that. And then the second review introduced the concept, and he called it stimulus trafficking. And I think that's where it went undone, right? Because you put trafficking next to stimulus and people were thinking of trafficking. But that review was about bias, right? What we now call bias. Yeah. And 1997 was another seminal paper that I think people have forgotten. That's Bill Clark and Kelly Berg. They worked with Paul Leff and they looked at the 2C receptor. And all the predictions of a two-state model will predict for biased agonism, they published it. That was a really, really important paper that I think not enough people give credit to. Because then later, Michel did his work and Bob did his work. And of course, Michel had a classic paper in 2003 in PNAS, and Bob's done all that work. And in fact, the word biased agonism, it was coined by Bob, if I'm not mistaken. So that's what stuck.
I think so.
But that's where that came from. And then, just again, like serendipity, then the analytical models that sort of started coming out for bias all happened around the same time. So Terry and I met, we're talking at a conference in Lille in 2009, and we both realized we were working on the same type of model. So then we ended up doing it together, and then Fred Aylett published a model in 2009 that's essentially the same as the one we did, but a lot of people had missed it, that he had published it earlier. I published it, and so, anyway, it doesn't matter. I mean, it's the same. David Hall's done work on this. Sue's done work, but it's just how things come together at the same time. And then all of a sudden, it's like, boom, they take off, right? But I think there was some early work that people often overlook now that everything's taken for granted. It's in the textbooks or whatever, right? Yeah, that's all I have to say about bias. I'm not going to go any further. That's all.
No, but it's funny because when I was writing my thesis, so I did my thesis on chemokine receptors, and so I had 3 papers in my PhD thesis, and my first paper was actually a collaboration with Michelle, where we're looking at using Brett biosensors on CXCR4 homodimers and CXCR4 CCR2 heterodimers. And then the second paper was mutations in the receptor in CXCR4, where we looked at signaling, but also dimerization. But the third one, which I have to say was my favorite, because He was on CCR2, and I used all of the known or potentially suggested 7 chemokines. And I tried to dissect which chemokine does what on which pathway. Was the one where I was hoping to see bias. I didn't. And that's, I think, the two Terry papers that you mentioned, I'm pretty sure were reviewed and included in my introduction for my thesis, because I do remember clear, and I wrote it in French, but I do remember clearly not using one single term for bias signaling, but it had the 2 or 3 that were in use at the time. And I explained that all of these mean this.
Yes, I remember having a slide for that 2005 meeting where I actually had an umbrella, and I had functional selectivity on the umbrella, and then underneath I talked about different mechanisms, desensitization, stimulus trafficking, and the other thing I recall, I actually reminded Terry about this. I'm pretty certain in the 1980s, Terry did use the term functional selective, but he wasn't referring to bias. He was, I think, talking about stimulus response differences between tissues and such and whatever, but it was used in a different context. And I know there's lots of other studies that I've missed and people that I haven't been necessarily. This is just my perspective. And also sometimes, semantics get in the way of good science. So don't be too precious about owning anything. Nobody owns anything. As I said before, the only thing you can claim in biomedical research is the start of a good idea. Everything else is done in collaboration.
Yeah, agreed. No, agreed. No, I think it's really, I still find it fascinating, whether it's bias or allosteric modulators or endogenous or natural, and the AGRP example that you mentioned. I think it was before we hit record.
It was before we hit record. Yeah. So the 2 points to make there. I mean, biased agonism is basically, it's all allosteric. It's allosteric turned inwards, right? So the modulator is the transducer, which is it could be a G protein or an arrestin or something else, and the same principles apply. That's bias, right? It's allosteric into the cytosol, and so it's like mirror images of each other. And that was in the AGRP. That was offline. I'm talking about where does the body make endogenous inverse agonists and why? And it's interesting once you start linking them together, rhodopsin, then you move to something involved in satiety, then you move on to something else, which are obligate heterodimers like the class C. What are they? Nutrient sensors, and then you move on. It's telling you something. It's telling you what's the most important thing in terms of us sensing the extracellular environment. And then so I always go to, that's my point. I always say, look at nature first. What is it telling you? And then take it from there, rather than trying to come up with if you've discovered something, then try to come up with something cool to call it. I just go back and see if it's already there,
Yeah. No, absolutely. I think it's really fascinating. And so Terry Kanakin just in February gave his class applying pharmacology to drug discovery. And Terry, you've seen Terry's slides.
Yeah. Oh,
and
yes.
Without Terry explaining, you'd wonder what is there. But I've really, really liked him, the way he's a great teacher, and to start with. And I really enjoyed, I think he had an mGluR 5 and 6 example. And they were curved. He was showing curves, and then he took the compounds and the receptors from that system, put them in another one, and the curves went into a complete other direction. And I was like, wow, isn't this cool? Where depending on which system you're using, depending on which assay you're using, getting back to the robustness of the assay, but also the expression levels of the receptor, you just get something else, which is really phenomenal.
Ask Nicholas Smith. So I think I may have mentioned it. I don't know if I mentioned it in one of the other podcasts or not, but we've got Nicola did her honors with me, and her project, we never ended up publishing her project, but it was on the 5-HT. I should revisit it. We took the cell lines from Bill Clark and Kelly Berg, where they had different expression levels of the 2C receptor, right? And in fact, the reason I started getting into the 2C receptor early was because I had chatted to Michelle, and I knew the story was coming out about, the bias story that he was going to publish with the beta receptor, where, and it was Michelle that said to me, look, take some of your antagonists or your inverse agonists or whatever, and look at MAP kinase pathways to see if something's going on there. That's how I got to actually first to map the 2C receptor to map kinase. It turned out it wasn't being done. And I'm looking around, and no one had done it. So we had to do that first. And then I started looking at these things, and I didn't see what Michelle saw. But when Nicola was in the lab, I said, let's just look at a whole range of ligands, just what the effective receptor expression does to the same set of ligands, agonists, neutrals, and inverses, right, in the same cell background. And the pharmacology completely changed of what we thought they were with the expression level, 2 different expression levels. That just blew my mind, right? And there's all these theories we were having at the time about what it could mean, but we didn't follow it up, and that's still. But I saw it. Nicola saw it.
No, that's phenomenal. And speaking, you were talking about allosteric before and bias signaling. The way Terry taught this class, which got me thinking. Now, when I think about proteins in general and protein interaction and its consequences, I always think about allosteric modulation, whether it's one or the other, one partner to the other. But you're right. You can have a ramp that could be modulating. You can have a G protein. You can have a GRK. You can have a beta arrestin.
The ramp is how Patrick and I started collaborating, right? So I got back from my postdoc. I met Patrick. He was doing all this cool work on ramps, the amylin receptor and everything. And that's how we started talking. Can we model ramp receptor interactions or whatever in terms of allostery? And because I was thinking this got to be allosterically modulating the phenotype, and then was either modulating the binding or what at the time the paper we published in 2003 we were showing was also differentially changing signaling of class B receptors more so than people had appreciated. So there was clearly that must mean there was some possible bias happening there as well. But that's how, So that's how Patrick and I started. I was the allosteric guy and he was the ramp guy, and we said, all right, let's go, let's see. It's fun.
What are your thoughts about allosteric modulators as drugs? Because I look around the landscape in the GPCR biotech world, and there aren't many companies that say we're developing allosteric modulators.
They are now. But this is one of the silly. I know. Let me gather my thoughts on this because this relates to something I mentioned earlier about pharma. The ones that sort of had been discovered serendipitously. There are a bunch of clinical trials, but again, a lot of them would have been discovered not knowing the kind of principles about what we know now. You mentioned earlier on, you want to be one of the beauty of allosteric, you can dial the drug to the disease. So you need greater degrees of cooperativity for a particular disease. Sometimes you need a little bit on the dial, sometimes you need a lot. I think a lot of the earlier the 2000, what's coming out of the 2000s and you're seeing trials, a lot of them have failed or crashed out. They weren't applying those sorts of principles. They were going for bigger is better, for instance. The second thing that happened at the time, a lot of companies got out of certain therapeutic areas that would have benefited from allosteric modulation. Now they're coming back. The best example I can tell you right now is, because it's close to my heart, psychiatry. M4 PAMs are now back in clinical trials because there's M4 for psychiatry. Right? That's a classic example. So smaller companies like ADX, if you know, ADX was the guy that Roche started.
That's exactly the one I was thinking about.
Well, I was in the early days. I have ADX stories too, because they flew me over to Switzerland. It was great. I got to be on their SAB for a while. I consulted, but it was in the early days. And I remember this was around the time we put all these templates in, started putting them into GraphPad Prison. There was one day where they put me in it was funny, but I was in a room and it was basically, I'd be meeting with group after group and they'd just be giving me data, analyze. So I'd be analyzing some of their data but doing that was a useful experience for both of us because we could see if things couldn't fit and I could point out why. I'd say, well, if there's too much noise in this part of your curve or this part of the assay, and that's why you can't get so you've got to clean this bit up and stuff like that. But I also saw what they were doing, and I could see things like, and then I went to sort of doing similar things, consulting for larger pharma. I'd have similar scenarios, but I could be talking to the screeners. I'd have a certain issue. This is, of course, post 2000. The ADX was earlier, post 2010. When the lab we were doing a lot of work in a lot of different areas, and I knew more because I was working at MIPS, so I knew a lot more about PK, ADME, formulation challenges, tox, or whatever, screening, computational biology. We're doing structures then as well, so I could then meet and talk to the different groups. So I had a meeting in one 2-day visit, I remember it was a Pfizer. It was fantastic because I got to talk to the screeners, and we'd have a different conversation, and then I talked to the chemists, and we'd have a different conversation, and then I talked to the structural biologist, and we'd have a different conversation, and so on. And so then I talked to the safety pharmacologist. So I'm not, and then we'd have a different conversation. That's why I think at the time, if people didn't understand what you should be looking for, why would you want an allosteric modulator if you can get an orthosteric agonist or an antagonist? And if you can't answer that question, you're jumping on a bandwagon and you're going to choose the wrong thing.
Yeah.
And right. And a lot of the earlier programs just weren't applying that lens, and or the idea that structure activity relationship alone, you're going to require at least one more number. You're going to require something that measures cooperativity, because it's different to affinity per se, even though thermodynamically you can show how they're linked or not linked. And just that alone was a language and a transition barrier, just so, getting the dialogue between the biologist and the chemist right. And then once you get that happening, then the next step is, all right, but what am I looking for in the disease state? What's the disease state calling for? Because the other thing that I talk about is tone-specific pharmacology. I mean, the adenosine paper that we published a couple of years ago was a really good example of that. It's disease specific, because what the modulator does to the agonist, the agonist does to the modulator. They're reciprocal. So you take advantage of the existing tone. Do you want to restore tone, or is tone elevated, say in a disease, or is elevated tone part of the reason you got the disease? Because what if inflammation? What if you got all these mediators in the soup that normally you wouldn't have, and they're endogenous allosteric modulators, but they cause disease, or allosteric autoantibodies? That's a whole other field as well, and so on and so forth. So it's about promulgation of ideas to different fields. That's the other thing I've been fortunate to do because of this. I've gotten to speak at different types of meetings. I've spoken at cardiovascular meetings or gastrointestinal meetings, endocrinology meetings, inflammation keystone about this very thing. It's more about getting the concept out there and then letting the field do what they do with it, and also me learning about their stuff, right? So
exactly, and learning these languages, because you mentioned that 2-day visit at Pfizer where you spoke to the screeners, to the chemists, to the admin talks, to the in vivo. All of those teams speak a different language. So in your case, you had to speak all of those languages.
There is no way in the world I could have. What year is it? 2020. There's no way. I'd say 15 years ago plus. There's no way in the world I could have done that, even as I was working out stuff as I was working out, because it goes back to knowing what you know and acknowledging what you don't know. And right. But that was fascinating. But that's why I think now is the era, I hope, especially with the smaller, that's a lot, for example, how we're working with Septerna. Now is the era where you would expect more of the rational design principles that people have uncovered in different groups about allostery, whether it's through the chemistry or the way to set up an assay, or the way to analyze the data, or the way, now that we've got the structures coming out as well. You'd hope to see more rational designed approach, allosteric drugs. I refer to them as allosteric medicines on purpose. And in fact, I actually mentioned that to Jean-Pierre because he was floored when he came to Australia, and I think I mentioned and saw how much the GPCR field were doing. And he himself said to me, "I love the word allosteric medicine, as opposed to an allosteric drug, because it's like the end goal is in mind, and it's a medicine." But the thing that we need to superimpose on that, and that's the hardest one of all, is obviously the disease context. Right? And you work backwards, okay? And that's the same with bias, right? The disease context. And so what sort of, where do you want the dial set, right? And then you've got to work backwards. And also, do you have appropriate enough models to be able to measure what the dial is doing? All right, that's where we are now. That's a challenge. That's why I'm also working with clinicians. And so selectivity is one thing, but what do you want? You don't want to turn acetylcholine into a toxin by boosting it by 1,000-fold. But early muscarinic PAM programs, that's exactly what they were doing. They were getting their most robust PAMs, and then they were seeing, tox essentially. You might see convulsion, because it's almost like you're overdosing them on your acetylcholine because your cooperativity is too high, right?
Yeah.
So it's stuff like that.
It's all about and you're right. I love the word dial, because you really want to dial it in You just have to, well, just, and I'm using air quotes here, you just have to know what do you need, what do you want to dial, and what's the clinical, what's the path to that clinical
That's right.
medicine.
Yes, that's right. So at least at the structure, activity, relationship level, you should be looking. Because I remember asking some of the folk in some of these companies, and then they dropped the programs for these reasons. But I actually said, well, in your screens, have you ever encountered PAMs or modulators with alpha factors of say 10 or 30, not just 100 or 1,000? But they go, no, we never actually looked for those. I said, they're probably the ones you would have needed. And then if you took those and then made them more drug-like, you wouldn't have the problem. You wouldn't have dropped the program. And I think more people are appreciating that now. Certainly, Craig Lindsley and the team, they know this. So if you look at, and I think we discussed this last time about where do you need, calcium sensing receptors only a little bit, mGlu's only a little bit, and so on and so forth. You really need to think about how much you want to dial. Otherwise you're going to end up with, well, as I said, imagine if the benzodiazepines, which have alphas of 3 to 5, I mentioned it last, imagine, but they're nanomolar binders. Imagine if they had alpha factors of 100, you'd kill the person, right? You'd boost GABA by 100. Well, what are you going to do to a person, right? So look at what the biology in the disease is telling you what you need, right?
Exactly. Did I hear you right when you mentioned allosteric oral antibiotics? Maybe I didn't.
Sorry, no. Oh, no. It's
with the
what
inflammation.
I said. Auto antibodies.
Oh, auto antibodies. Okay. Okay.
There's been some work from, was it from, there was some work on this. I'm trying to, was the group in Canada again. Oh, I apologize. I've got to draw a blank, but not just that. There was there was an example of the, was a beta 2 receptor auto antibody, I think, an endogenous allosteric modulator affecting meningitis. I'm trying.
Yeah, I'm blanking too, but I have a vague memory of seeing not too long ago.
I'm trying to remember the work, but also there's earlier work with Sjogren's disease, which affects muscarinic receptors. There's evidence that one has, and it's more prevalent in pregnant women, it turns out, they're allosteric autoantibodies that cause things like xerostomia and things like that by the M3 and the M3 receptor. Chagas disease, which is another one that has been suggested that, so that's you see that it's a tropical, I think a parasite. You make some, I can't remember where it comes from, but I think it's like that. But that's been suggested that we have allosteric autoantibodies are raised. How do you get Chagas disease again? I'm trying to remember. Anyway, that's what I mean. I've forgotten so many ththings, but to the beta 2 receptors in the heart, and I think also muscarinic receptors. And then there's more. I'd suggest go back. Again, the review that we wrote in 2015 in JPET on endogenous allosteric modulators, we have a section on autoantibodies in there as well. Emma van der Westhuizen is the first author on that one. So, and that's an area. The reason I find that area really interesting, and when I spoke at the inflammation meeting and also some of the cardiovascular meetings, I said, so those conditions theoretically is where maybe a neutral allosteric ligand, and now is the preferred therapeutic approach, because you don't want the PAM and you don't want the NAM, you just want to block a bad behaving endogenous allosteric ligand. So that's where you need a NAL. NALs aren't just tools, they can be drugs, right? And that's the other point so when you're doing that, in addition to when you're looking at your screens, so you should be looking for low-level cooperativity factors as well as high-level cooperativity factors, whether it's a NAM or a PAM, but you also think about your inactives in a given scaffold series, because some of those could be NALs, and you can just do a simple competition experiment with a PAM or something to find out if they are or not. They're useful A as probes because you can, but B, they could be medicines, and there is a medicine on the market already, and again, it's in the GABA field, flumazenil. Flumazenil? That's a NAL. That's what we call the NAL now. That's used to reverse benzodiazepine overdose. So that binds to the GABA A receptor and blocks benzos from binding to the same site, but it does not modulate GABA. So it's out as one, and it's used. It's out there. It's used to reverse benzo overdose. So there is one on the market, but people hadn't realized because we weren't calling it NALs. So that's why you got to look around. So Yes, so I think NALS is another interesting area. I guess we're going on a really big rambling talk now about allosteric medicines.
Yeah.
That's what yeah, getting your mind around it, getting the dialogue between the different groups right, setting up the assays appropriately. But the end goal is, what's the disease context? Do we have the models for it? What do we need? And once you accept,
Exactly.
I need a boost of 10. Well, that's great, because that's what Jim Black was telling me back in when I met him. He was saying, I think I mentioned that last time, dial it down and you lose he thinks the agonism would be bad. He said, dial it down, and that's what, and then you'll get smaller cooperativity factors, but they're probably all you need. And then you'll avoid, on-target tox as well if it's too high. And that's, I think, the case. So programs are being shut down, and now some of them being reopened. So now we're going to start to see if it pans out or not, because and with the structures as well, now people are trying to rationally design allosteric modulators, and it's 2024.
Yeah.
Yeah. Yeah, I told you, 2002 was that review, and yes, I'm one of the old folks now.
Oh, yeah. No, I think it's really phenomenal. And when you think about it, I actually, someone asked me this week or last week, I can't remember, if there's any endogenous allosteric modulators, and I said, honestly, I don't know. And now I do.
Read that review, the JPET 2015, endogenous allosteric modulators of G protein coupled receptors. And we talk about things like, lipids and ions and what we knew at the time, about peptides and proteins and, accessory proteins, and going all the way to autoantibodies. But a lot of that, not a lot of it has been studied mechanistically enough. But if anyone's interested in any of those aspects, you should be looking at them. That way. The iron effect, the sodium effect is very well known, and such, but there are enough examples there. And I think inflammation in particular is the area where this, and resolution pharmacology, all that sort of stuff, not just in anti-inflammatories, but pro-resolving mediators as drugs. That's a fascinating area where the soup changes dramatically. And then what's in the soup? That's way beyond me, but if you're interested in that field and you bring an allosteric lens to that field, I think it's going to be, if you've got the right assays, it'll be really, it's all untapped. That and the autoantibodies, because that's right, there are other examples of preeclampsia. There's another example where I think there's autoantibodies to angiotensin 81 receptors, I think, all pretty sure. There've been some examples of that can cause that. So again, a now for that, right? So yeah. Sorry.
It's no, please. No, I think it's really great. And I think again, you made a great point when you said you need to run the assay properly, and you need to have the right assay to run this. Because I think there's been such a focus on orthosteric sites for such a long time that when you're looking for an allosteric effect, you need to be able to see, you need the right lens basically to see it and measure it properly.
Yes. So and hopefully we're there now, in the sense that, people have accepted that these things are real, and hopefully the driving structure activity work, as I said, that can, appropriately or enough, add one more number if you can, and then the rest is assay design, and also, but the disease context is everything, and animal model, and the model, the predict, and who knows what AI is going to do to all of this, right? So I'm not. But here we are. Yes, it's in the textbooks now, so I think people believe it, but it's what they do with it that matters.
I agree, and absolutely. Especially if you have a path, if we're thinking about medicines, allosteric medicines, which I actually really do like, then you need, not only you need to be able to, find these and measure this in cells, but you need a path to the clinic.
That's right. So you need to be incorporated, and we know enough now that you need to be incorporating things into your target product profile. You need to be incorporating things into candidate selection matrices, before you choose a candidate. I mean, so look, I'm looking back in the early days, how naive we were, but we were talking about SAR, but now we've progressed to the point of what is the TPP, right? And if you can't answer what TPP is for an allosteric modulator that you want to turn into a medicine for a particular disease, you haven't done your homework. That's it. Exactly. I'm not saying I'm saying you haven't done your homework. That's the way you need to be thinking about it. why would you want an allosteric modulator for disease X or a biased ligand? And then, what does a TPP look like? And that should, and then work backwards,
Yeah. Lots of fun.
Yeah.
Lots
That's the
of
future.
fun. Yeah, I think
Yes.
so too. Lots of fun things to think about, to work on. I think there's
But
a
just
bunch
to
of
clarify, in case some people listen to this, I think most people know what a TPP is, but I mean target product profile for those that don't know
yes,
what
you've
TPP.
used it. Well, I have to say that the first time I heard the word TPP, I had to Google it.
Yeah, target product profile. Yes, that's
Profile,
a very
yes.
target product profile. And sometimes what we've used it for is when some of our partners, when you have good partnerships, with a farmer or our collaborators and such, we say TPP could also mean target partner profile, but target product profile.
Product profile, yes.
I don't know what a TPP is. Yes. And that's key for drug development. You've got to have a TPP when you're launching a program.
Yeah, great. Arthur, thank you so much for all your time. I really appreciate it. I do realize that it took a while to get here, and I really appreciate you coming on the podcast and being so generous with your time. It was so much fun.
It helped me. It's good to talk science again and not just be burdened with admin. Yes. So, and good luck with the book. I'd like to see how that.
Thank you. I need to make time now for at least sitting down and putting down an outline, because this is an episode where we talked about allosteric modulators. I've recorded 1, 2, maybe 3 episodes with Richard Permont, where first one we talked about his career, and the other 2 was history of G proteins, a little bit about GPCRs, where Richard had really dug in and you had the references and everything. But I think it doesn't give the now PhD students and postdocs a full picture of where do these concepts come from, what were the seminal experiments, the observations, and how we got to where we are. So I need to.
Well, and I'll just want to put a caveat on this. This is to the best of my recollection and the best of my knowledge as I was studying as a student and such. I know there's a lot of stuff I've missed, and people will have different stories. And this was off the top of my head. This is not trying to. But there's a lot there, and then it's just for everyone else to build on. But I hope there's enough interesting stuff in there, then people can dig in and find more. Yeah,
there is, and it's funny because I see your your Septerna logo on there.
Yeah, I'm wearing. Sorry, yes. And Sergeant Pepper's Lonely Hearts Club, the other bit, yeah.
And the reason I remember this is because I was at a GPCR biotech meeting, the 3rd GPCR Drug Discovery Summit, and I was there with a couple of my colleagues, but also with a couple of people from Septerna as well. And we were just talking about that episode with Richard, where we were talking about Al Gilman, and they were trying to reproduce one of their assays, and it turned out that they thought it was the water. So somebody drove up to Virginia to bring the water, and it turned out that it was the aluminum in the tubes that they were reusing that allowed the activation of the G protein, the aluminum fluoride.
Actually, I think this is a good point to end this on, because it's something that I learned from Jim Black. It's something that I try to do every time someone new joins my lab, and it's not real if it's not reproducible, right? That's right. So we always try, even anything that we get from anyone, any compound or anything, the first thing we try and do is we do reproduce the key experiment or the key finding. That before we go, don't take it for granted and then you just go, right? You got to be able to reproduce at least the key elements of something, and that's key. And sometimes it boils down to things like this, but lucky for them. But I still make that a rule to this day. And Jim was always the same. He always, any student he would take it in his lab would say, first thing you do is go read a paper or an interesting thing, reproduce it, then you're ready to start your project, basically. So
love it.
I think that's a good lesson.
I think so too. I have a brief story about that. I had a colleague in my PhD lab who was a master's student at the time, and he spent the entire summer trying to measure the cell surface expression of some kind of a protein, only to realize at the end of the summer that he never resequenced the construct that we got from a collaborator, and the N-terminal, the transmembrane part of the construct was missing. So every time he would throw away the media, he would throw away his protein.
Yeah, you're not alone. I think that some of them had to go back and resequence things. In fact, they're doing it again now, but that's whole. I don't want to go into that whole other story.
Yes. So you double check, you sequence, you make sure that everything is properly done and reproducible.
Yep.
All right.
Science.
Yes. So much fun. Don't go anywhere. I just want to say thank you again, and I look forward to. But actually, you mentioned that you're going to be next year at the GRC in California.
That's the intent. So it's the February. I think February.
February, yes. I already blocked my calendar. I know registrations are not open yet. I'm gonna lobby Martha and Silvio to be there. The family is ready, so I hope nothing changes and we get to meet in person.
Oh, we will. I mean, I'm looking forward to my first trip in years. May, I'm going to the ASPET meeting, and so that's be a time to reconnect with some friends I haven't seen in a long time. So hopefully I'm back on the wagon again, hopefully.
Fantastic. Thank you, Arthur.
Thanks, Yamin. Take care.
Hello, everyone. This is Yamina for Dr. GPCR, and I am happy to welcome back Dr. Arthur Christopoulos for an addendum. This is the first time we're doing this. Arthur, welcome back.
Well, thank you. We've got to stop meeting like this, I have to say, but thank you for giving me the opportunity, because people don't realize, obviously, that we've recorded these conversations at different times, and the last podcast that we did was, I think it will be number 3 with me, right? And it was a Sunday morning, if I'm not mistaken, early for me, and we were talking about allosteric, and we got to part of the conversation where we were talking about allosteric auto-antibodies and endogenous allosteric modulators. And I kind of got carried away when I was talking about some of their work. And there was one study that I mixed up and conflated. Like, I was talking about auto-antibodies in the context of meningococcal disease and bacterial infection. And I realized straight after I finished, I emailed you and said, no, I got that wrong, because it was actually an example of biased allosteric modulation. So I want to correct what I said, because I didn't attribute the group. I was trying to think of who it was, and I remember later, straight afterwards, it was Stefano Marullo's work. Yeah. That's who I was wanting to talk about. And they had done this fascinating work, and I remember seeing early versions of that at conferences, and that's what the allosteric thing triggered in me. They basically showed that the meningococcal bacterium, or however you pronounce it, they have hair-like filaments called pili or pili. I don't know how they're pronounced yet, but they're these protein structures. And basically, those pili or pili interact with N-glycan regions of the beta-2 adrenergic receptor to cause a mechanical stimulation of the receptor, and that leads to selective biased arrestin recruitment. And that leads to changes in endothelial cell permeability as part of the infection process. And I remember seeing that work, and I was fascinated. And I asked Stefano at the time, and obviously, because of the nature of the interaction, it had to be allosteric. And that's why, and it was, and it is. It's essentially, you have a bacterium, part of a bacterium, allosterically interacting with the N-terminal region of sialic acid residues of the beta-2 adrenergic receptor to mechanically promote biased allosteric modulation of the beta arrestin pathway as part of the disease process. So that was a study that I was referring to, and I apologize for drawing a blank at the time. So I wanted to correct that and point that out too, because it's a fascinating example of allostery.
It is a fascinating example, and it goes back to the fact where we were talking during the podcast recording, that the GPCR itself and anything it can interact with is an allosteric interaction, other than the natural ligand.
That's right. I mean, we said the G protein is the obvious one, but in fact, even in this work, Stefano, if I'm not mistaken, I think another mechanism that is involved in this process is, I think it's CD17 or one of those co-receptors has to also heterodimerize, I think, with the beta-2 adrenergic receptor as part of this process when it's interacting with the bacterium. So you do have biased allosteric modulation of a GPCR via mechanical means, and then a heterodimerization process with another receptor. I think that's where the work is at now. So it's fascinating.
Functional selectivity.
Yes. So there you go. Anyway, I just wanted to say sorry, Stefano, for forgetting to talk about that work in properly. So that's That's the addendum.
Thank you so much, Arthur.